Well, welcome back to Biotechnology. We're talking about the PCR method, and we've just finished talking about the overall process and all the steps involved. And now we're going to look at some types of PCR and modifications that have been made over the past 20 years or so. There are several types discussed in your book. I'm going to focus on the three types, and particularly because these are all involved in or being used right now for the COVID-19 testing and for many other disease diagnostics. So we're going to look at quantitative PCR. The other name for this is sometimes called real-time PCR, but I like to use the word qPCR so we don't get it confused with the RT-PCR, which is reverse transcriptase. So as we might guess, quantitative PCR lets us determine how much target DNA is present. When we do PCR, we can see a band on a gel. We can sort of estimate how much, but we really don't know precisely. So this is a modification that lets us get much more specific. The second modification is called reverse transcriptase PCR, and this is where you start with RNA. So if you're going to amplify a gene in PCR, you have to have DNA. So if you have a virus that's RNA, how do you amplify that? So the the retroviruses have an enzyme called reverse transcriptase that take RNA and copy it to double-stranded DNA that's called cDNA. And this is something that was mentioned earlier in our book, but we didn't emphasize it. So I want to mention cDNA, which stands for complementary DNA, which is complementary to the RNA. So the COVID virus, for instance, the SARS-CoV-2 virus is an RNA virus. So there's a step where you have to copy that virus genome to DNA, and then you can amplify it in PCR. And then finally, a very exciting innovation is what they call isothermal PCR. So normally PCR requires that cycling through many different temperatures. Isothermal PCR actually allows you to amplify at a single temperature. So you don't actually need a thermocycler, you just simply need a water bath to warm up the reaction. And it gives results much faster. So any of these methods, conventional PCR, or these other methods, minimally require probably 3 to 4 hours. A lot of the isothermal, also called LAMP, which stands for loop-mediated isothermal amplification, and I'll show you a picture of that in a second. Anyway, many times these will work in sometimes 30 minutes or less. So it's a really neat innovation if you're trying to do point-of-care diagnostics or get an answer really quickly. So the first type we'll look at is used to be called real-time, but like I said, I prefer the qPCR, quantitative PCR. And in this type of PCR, your nucleotides have a fluorescent dye that's incorporated. So as the nucleotides are getting incorporated into your product, you build up fluorescence. And so in your plate reader, or in your thermocycler, you have an optical reader that scans all of the wells and looks at fluorescence intensity, and it monitors this over time. And so this allows you to actually watch the growth of the curve. So you really get that, you get to watch the exponential growth curve develop. And so a lot of detection systems for human pathogens, and these could be in blood or in urine and other kinds of samples, these are really commonly used. So this is the kind of result you get in your quantitative PCR. Instead of having to wait and run a gel and look for a band, you actually get these cycles that are building up over time. And so what you do is establish some baseline threshold, and you say, well, if I get to this many copies, then that indicates I really do have a real sample. And so what you can see is if I have a lot of DNA to start with, I don't need very many cycles. So for instance, if this is my baseline, this sample I got to my target in about 16 or 17 cycles. Whereas if I look down here, this one took maybe 36 or 38. So the number of cycles that you take to get to that positive is then inversely related to the amount of initial target DNA or whatever sample you started with. So if your target sample was really high, then it'll take you less time to get to this point, whereas if it's low, it'll take more time. So this is sometimes called the CT or cycle threshold value, and that's something that's been reported. Some of you were listening to This Week in Virology, they were talking about cycle threshold and sensitivities of different methods. So coupled with that, then, if you do have an RNA sample, so if you do have a virus that has an RNA genome, you'll use the enzyme reverse transcriptase first to copy that RNA into DNA, and then use that complementary DNA, that cDNA stands for complementary, because it's the complement and it is double-stranded DNA. So that then becomes your template. And so in your exam or in your mix, you'll have a first cycle that'll hold at a certain temperature to let that reverse transcriptase reaction take place, and then you'll go on cycling. So as I said, this is used for a lot of RNA viruses. So the SARS-CoV-2 virus that causes the COVID-19 disease is one of those examples. So the last method is a really exciting method, and I have a couple animations to show you, a couple videos in the links. So isothermal or LAMP PCR refers to loop-mediated isothermal amplification. It's carried at a single temperature rather than having to go through that high, medium, low. And it runs much faster because you start to get a chain reaction off of multiple sites. So what you end up doing then is having these little loops that form. And what's really neat about this process is as the loops form and you're incorporating nucleotides, you release phosphate ions, so PO3 minus 2, so it's basically phosphoric acid. So the very newest versions of these are actually just detecting the acid that's being released. So as this phosphoric acid's released in amplification, it changes the pH of the sample. So you don't even have to look for PCR product at all. You're actually just looking for pH shift, and you get a simple color change. So it's super easy to read. It's really exciting because the equipment that you need is very minimal. And this is some of the latest diagnostics for COVID. If you've heard about the Abbott Labs diagnostics that's being used by, I think it's two T's, Abbott Labs, where they do a lot of medical diagnostics, but this is one of the ones they've developed for the SARS-CoV-2 virus. So anyway, super neat. And again, you get the result in very rapid results. So sometimes, like I said, 30 minutes or less in many cases to get that answer. So as we touched on before, PCR is probably one of the most widely used techniques in the modern biology lab. I don't think of any labs that don't need this for at some point. Ecology labs, wildlife biology labs, it doesn't matter, and certainly in clinical labs and in applications like forensics, there's a lot of places where we're using PCR to identify people or remains or suspects, and we're going to do a simulation of that in lab. So many applications, and of course, just basic research. That's really important. So in basic research, we can use the PCR product. We can do DNA sequencing. We can actually identify organisms. There's a technique called barcoding, something we'll do a little later in the semester, where you amplify a piece of an important gene and then get that sequence, and you compare that then to what's known in the databases, and you can identify plants and animals and all sorts of things. And you can also clone your gene. So you can actually put your gene out of your PCR product into a cloning vector and then get it to produce more of the gene, or get it to produce the protein that's encoded by the gene. So it saves a lot of steps in the whole cloning process, and it's just revolutionized that aspect of basic biology. As we mentioned in medicine, PCR-based diagnostics or molecular diagnostics have revolutionized the way we diagnose. So traditionally, you would have to take a sample from a patient, cultivate the microbe. That minimally takes you a day, but many times it takes you multiple days to do the tests and isolations. So you either directly cultivate and identify the microbe, or you wait for an immune response to the microbe and you look for antibodies, and that's still a very important way to diagnose infections. But that's also delayed by many days or weeks because you don't get antibodies typically until 7 to 14 days or longer. So what's valuable about PCRs is they are highly specific, and you don't need a pure sample. So in many microbiology techniques, you have to get a pure culture to go about your identification, but you can take a mixed sample. You can take blood, sputum, you can take urine, anything you want, as long as you have the primer. So you do have to know what you're going after. That's the important thing. But you can get the answer within hours from the time the PCR started. So whatever time it takes to get to the lab and set up the reaction. But once you get it going, you get that answer with hours versus the days to weeks on these other methods. And it can be applied to infectious diseases, but there's also non-infectious diseases, so genetic disorders, certain types of cancer mutations. There's a wide range of things you can look for. So pretty much anything that's in DNA or RNA, you can find it with PCR. So just a little bit more about the SARS-CoV-2 diagnostic. So it's using a combination of the reverse transcriptase along with the qPCR. So it's quantitative reverse transcriptase PCR. And so the primers are designed for the SARS virus. They selected primers from regions of the gene that are in the nucleocapsid. And you want to find things that are conserved because the virus will go through mutations. So you want to find something that even if the virus goes through a little bit of point mutations, your primers are still going to bind to that virus. So this is some of what the data looks like. This is from one of the CDC sources. So you have kind of sort of background noise in your amplification, but real amplification then actually goes through and makes these very nice curves here. And again, you can see your cycle threshold. So you can see a sample here became positive at cycle 20, where this sample over here didn't become a positive until cycle 29. So we'd say this sample here had more virus because it took fewer cycles to get to that threshold than this one here, which took longer to get there. So another application of PCR is used in agriculture. We have many organisms that have been modified through genetic modification. And that modification we'll talk about a little bit later, but it usually involves the soil bacterium Agrobacterium tumefaciens. And this invades a variety of plants and causes crown gall. It carries a plasmid called the TI, or tumor-inducing plasmid. And so that plasmid's been engineered to contain specific genes that can then incorporate into the plant tissue and give it some new trait. And so the traits that we're interested in are things like resistance to herbicides, so we can spray fields for weeds and our crop doesn't die. Resistance to pests, so that if the pest is trying to feed on it, there's a toxin in the plant now and it will kill that particular pest. And things like drought tolerance, frost tolerance, and actually an increased nutritional value in some cases. So all these are things that have been engineered into several different crops, and we'll talk about those a bit later. So if you have primers to detect the specific GMO genes, you can actually detect those genes even in food products. So an experiment we'll do a little bit later is we'll take a variety of foods. We'll have a non-GMO food and then food that we believe is genetically modified. We'll do a variety of PCR reactions. We have some nice controls built in, and we'll look to see, in fact, if they do contain a band corresponding to a GMO gene. So something to look at down the road. So in forensics, forensic scientists use biological samples, and this could be, again, anything from a crime scene. It could be saliva, it's frequently blood. And what you're trying to do is rule in or rule out whether or not your suspect was present at the crime scene by looking at these biological fluids. So what we tend to look for, what the FBI and other groups interested in this kind of testing, are what they call short tandem repeats. And short tandem repeats are regions in the genome that vary in individuals, and there's a variety of alleles in the population, but each of us only carry 2 copies of those alleles. And because, again, we're using PCR, you can get something that's done very quickly and also allows you to discriminate within individuals very quickly. So this is a quick example, would be the same sort of idea, but looking at paternity. So this represents the allele ladder. So this represents, in the population, you could have a large number of repeats or a few numbers. So we'll just call this 2, 4, 6, 8, 10, and 12 repeats. So this represents a ladder of potential. And so mom here carries an allele for number 10 and number 6, and dad carries 10 and 8. And so what you can, the father is 8 and 4. And the child carries 10 and 8. So what you can see is a child's going to have one allele from one parent and one allele from another. So it matches mom at this point, it matches dad at this point. So based on this set of markers, it looks like this child came from these 2 parents. So that's kind of how you determine a match in paternity. In looking at suspects, what you're looking for is a complete match, of course, between the crime scene and the suspect's information. So much of this has been automated. This is a sort of gel that would be run using the FBI, what they call the CODIS system. And we'll talk a little bit more about this later. And this actually looks at 13 different loci. So it's quite a complex pattern, but this is just one example right here. So what you can see again is your allele ladder in this lane, and these represent different individuals and which alleles they carry. So what we can see is this individual here actually matches, if I look at this lane, and so this is my 2nd lane and my 4th lane, they actually contain a match. Whereas the others alleles are at different locations. So this just shows you kind of one system, but you would do this at 13 different loci. And so the probability that anyone would match at random is very low because you get so many different possibilities through all of these different alleles. We can also use PCR to look at human migration, human evolution. So a couple different types of DNA that's looked at is the DNA in the mitochondrion. And this mitochondria DNA is circular, just like bacterial DNA is circular. And if you look at human evolution, as humans and as mammals, when sperm and egg unite, all of your mitochondria come from your mom. So there's a maternal inheritance of mitochondria. So you can trace back the human evolution, and they call this the mitochondrial Eve, but probably not one single individual, but at least trace the lineage back to the origin of where the earliest people were found. And most of this work is done in Central Africa. And you can do the same strategy with the Y chromosome because boys always inherit their Y chromosome from their dad. So you can look at that lineage and those sequences. So that's another way to apply this. You can look for specific markers within one type of DNA and track that through different populations. The other type of repeat sequences, so we have these short nuclear or short tandem repeats. We also have short interspersed nuclear elements called signs that exist throughout the population, and these are inserted in the genome multiple times over. And so the ALU sequences have been inserted millions of times really in our genome, and you can see hotspots of location. So these are in green. The ALU genes are in green, and you can see different chromosomes where we have a lot of ALU sequences, and some where not as many. And so you can compare these ALU insertions in different human populations or ethnic groups throughout the world. And again, look at migration, look at paternity, look at relationships, and so forth. And finally, we use PCR in wildlife conservation. You can use the same short tandem repeats to identify different animals. So you could apply it to even just eggs. So sturgeon eggs, which are used for caviar, you can type them into where their source is. If you have just meat from animals, so muscle tissue, you can distinguish legally caught whale from illegally caught dolphin. You can also identify teak and mahogany, which are valuable woods, and these are many times been illegally removed. And you can identify remains of animals. You can even look at things that have been dead quite a long time, things from ivory carving and rhinoceros horns and even gallbladder. So some of these are things that are thought to have medicinal value and they're illegally harvested. And so that we can identify whether those have been kept. You can also look at a variety of fishes. The other thing we'll talk about later is you can look at grocery store fish to see whether it's labeled properly, because sometimes we know that hasn't been the case. So I'm going to stop at this point for lab this week. We're going to do the lab on the short tandem repeat PCR analysis. And this is a fictional crime scene. And so I'm going to have you read, and actually I think it's up to page 209 or 210. So there's a bit of reading on the methods there. And so I'm going to set the stage in the fact that you're going to be a forensic investigator. We have collected DNA from a crime scene. Of course, this is all simulated, but we've collected DNA from a crime scene, and we have suspects from our samples from 4 suspects. So your task is to amplify the DNA from the crime scene and the 4 suspects. We're going to look for a match between the suspects and the crime, and we're going to see which suspect can be linked to the crime scene. So my challenge for you is to have a scenario. What was the crime? So we're in the month of October. We are getting close to Halloween. You can devise a variety of scenarios for that. So I'll let you think that over. And then some things to get out of this chapter and things to put on the quiz for this week. Of course, what does PCR mean? What is an amplicon? What are the primers used in PCR? What is the target DNA? Where does that come from? What are the steps in PCR? So what is the denaturation step and how high does it have to be? What's the annealing step? What are some things that determine when things are annealed? What's the extension step? What is Taq polymerase? What's a hypothermal file? What is qPCR, RT-PCR? What is reverse transcriptase? What is the LAMP technique? What is SARS-CoV-2 and COVID-19? And what's the difference between the two? Okay, one is the virus, one is the disease caused by the virus. And then some questions. So what are the steps in PCR and their functions? What are the typical temperatures for each step? what are the components of a PCR reaction? So what are the things you have to have in the PCR reaction to make it work? And what are the differences between conventional PCR, qPCR, RT-PCR, and LAMP? How does the diagnosis, or why does the diagnosis of SARS-CoV-2 require reverse transcriptase in the qPCR method? And some examples, how's PCR applied to forensics, medical diagnostics, wildlife conservation, and in genetics, and just biology in general. So those are the things we're looking at. Please let me know if you have any questions, and I will see you in lab.